Hello everyone. Now we are going to learn about immunology. Immunology is a science related to immunity and related aspects. So in this session we will be having a short overview of the immune system, some aspects related to immune system of human, some early vaccination studies and historical perspective related to immunology, then vaccination, some important aspects related to humoral and cellular immunity, theoretical challenges faced by them in the sense scientists, then infection and immunity, some other aspects related to innate and adaptive immunity, phagocytic cells are a barrier to infection, some topics related to that, soluble molecules, then some aspects related to adaptive immunity, T lymphocytes, B lymphocytes in particular, then some aspects related to humoral and cellular immune response and antigen receptors of B and T lymphocytes. And antigen selection of lymphocytes causes clonal expansion. Okay, so some other aspects of this topic will be dealt later. So let's start learning about immunology. This is a very important lecture series related to immunology. So let's dive into the topic. The immune system evolved to protect multicellular organisms from pathogens. Highly adaptable, it defends the body against invaders as diverse as the virus that cause polio and the flatworm that cause histosomiasis. The immune system generates an enormous variety of cells and molecules capable of specially recognizing and eliminating foreign invaders, all of which act together in a dynamic network. Protection by the immune system can be divided into two related activities: recognition and response. Immune recognition is remarkable for its capability or capacity to distinguish foreign invaders from self components. The immune system is able to recognize molecular patterns that characterize groups of common pathogens and deal with these in a rapid and decisive manner. It can even detect subtle chemical differences that distinguish one foreign pathogen from another. Above all, the system is able to discriminate between foreign molecules and the body own cells and molecules, non-self discrimination. In addition, the system is able to recognize host cells that are altered and that may lead to cancer. So typically, recognition of a pathogen by an immune system triggers an effector response that eliminates or neutralizes the invader. The multiple components of immune system are able to convert the initial recognition event into a variety of effector responses, each uniquely suited for eliminating a particular type of pathogen. Certain exposures induce a memory response characterized by a more rapid and heightened immune reaction upon later attack. It is the remarkable property of memory that prevents us from catching some disease as a second time, and immunological memory is the foundation for vaccination, which is a means of educating the immune system to prepare it for later attacks. Although reference is made to the immune system, it must be pointed out that there are two systems of immunity: innate immunity and adaptive immunity, which collaborate to protect the body. Innate immunity includes molecular and cellular mechanisms pre-deployed before an infection and poised to prevent or eliminate it. This highly effective first line of defense prevents most infections at the outset or eliminates them within hours of encounter with the innate immune system. The recognition elements of the innate immune system precisely distinguish between self and pathogens, but they are not specialized to distinguish small differences in foreign molecules. A second form of immunity, known as adaptive immunity, develops in response to infection and adapts to recognize, eliminate, and then remember the invading pathogen. Adaptive immunity is contingent upon innate immunity and begins a few days after the initial infection. It provides a second comprehensive line of defense that eliminates pathogens that evade the innate response or persist in spite of them. An important consequence of adaptive immune response is memory. If the same or a closely related pathogen infects the body, memory cells provide the means for the adaptive immune system to make a rapid and often highly effective attack on the invading pathogen. The chapter introduces the study of immunology from a historical perspective. The highly practical or applied aspects of immunology are outlined, emphasizing the role of vaccination in the development of immunology as a scientific field and as an important aspect of public health. A bird's eye view of the common pathogens to which we are exposed is given with a broad overview of the process, cells, and molecules that make up the innate and adaptive immune system. Last, we describe circumstances in which the immune system fails to act or when it becomes an aggressor, turning its awesome powers against its host. That means some topics related to autoimmunity. So, historical perspective, we are going to learn about them. The discipline of immunology grew out of observation that individuals who had recovered from certain infectious diseases were thereafter protected from the disease. The Latin term immunus means exempt. This is the source of the English word immunity, meaning the state of protection from infectious disease. Perhaps the earliest written reference to the phenomenon of immunity can be traced back to great historian Thucydides. I'm not sure about the pronunciation, I'm sorry. Philosophian, the Peloponnesian War. In describing a plague in Athens, he wrote in 430 BC that only those who had recovered from the plague could nurse the sick because they would not contract the disease a second time. Although early societies recognized the phenomenon of immunity, almost 2000 years passed before the concept was successfully converted into medically effective practice. Early vaccination studies led the way to immunity. The first recorded attempts to induce immunity deliberately were performed by the Chinese and Turks in the 15th century. They were attempting to prevent smallpox, a disease that is fatal in about 30% of cases and that leaves survivors disfigured for life. Reports suggest that dried crusts derived from smallpox pustules were either inhaled into the nostrils or inserted into small cuts in the skin, a technique called variolation, in order to prevent this dreaded disease. In 1718, Lady Mary Wortley Montagu, the wife of the British ambassador in Constantinople, observed the positive effect of variolation on the native Turkish population and had the technique performed on her own children. The English physician Edward Jenner in 1798 made a giant advance in the deliberate development of immunity, intrigued by the fact that milkmaids who had contracted the mild disease cowpox were subsequently immune to the much more severe smallpox. Jenner reasoned that introducing fluid from a cowpox pustule into people, inoculating them, might protect them from smallpox. To test this idea, he inoculated an 8-year-old boy with fluid from a cowpox pustule and later intentionally infected the child with smallpox. As predicted, the child did not develop smallpox. Jenner's technique of inoculating with cowpox to protect against smallpox spread quickly throughout Europe. However, it was nearly 100 years before this technique was applied to other diseases. As so often happens in science, serendipity in combination with astute observation led to the next major advance in immunology, the introduction of immunity to cholera. So Louis Pasteur had succeeded in growing the bacterium through to cause bowel cholera in culture, confirming its role when chickens injected with the cultured bacterium developed fatal cholera. After returning from a summer vacation, he injected some chickens with an old bacterial culture. The chickens became ill, but to Pasteur's surprise, they recovered. Pasteur then grew a fresh culture of the bacterium with the intention of injecting it into some fresh chickens. But as the story is told, His supply of chickens was limited and therefore he used the previously injected chickens. Unexpectedly, the chickens were completely protected from the disease. So Pasteur hypothesized and proved that aging had weakened the virulence of the pathogen and that such an attenuated strain might be administered to protect against the disease. He called this attenuated strain a vaccine from the Latin. Vaccine means cow. Vacca means cow. In order of Jenner's work with cowpox inoculation. Pasteur extended these findings to other diseases, demonstrating that it was possible to attenuate or weaken a pathogen and administered the attenuated strain as a vaccine. In a now classic experiment performed in the small village of Poly-e-Fort in 1881, Pasteur first vaccinated one group of sheep with heat-attenuated anthrax bacteria, that is Bacillus anthracis. He then challenged the vaccinated sheep and some unvaccinated sheep with a virulent culture of the anthrax bacillus. All the vaccinated sheep lived and all the unvaccinated animals died. These experiments marked the beginning of the discipline of immunology. In 1885, Pasteur administered his first vaccine to a human, a young boy who had been bitten repeatedly by a rabid dog. The boy, Joseph Meister, was inoculated with a series of attenuated rabies viruses preparations. He lived and later became a custodian at the eponymous Pasteur Institute. Vaccination is an ongoing worldwide enterprise. The emergence of the study of immunology and the discovery of vaccines are highly related or tightly linked. The discovery, development, and appropriate use of vaccines remains a challenge to immunologists today. In 1977, the last known cause of naturally acquired smallpox was seen in Somalia. The red disease was eradicated by universal application of a vaccine that did not differ greatly from that one used by Jenner in the 1890s. The consequence of eradication is that universal vaccination becomes unnecessary. This is a tremendous benefit because the smallpox vaccine carried a slight degree of risk to persons vaccinated as well as to persons exposed to recent vaccines. There is a darker side, however, to eradication and the end of universal vaccination. Over time, the number of people there is a darker side, however, to eradication and the end of universal vaccination. Over time, the number of people with no immunity to the disease will necessarily rise. Yet one day, the disease will be reintroduced by unnatural means. In fact, smallpox is considered one of the most potent bioterrorism threats. In response, new and safer vaccines against smallpox are being developed. A milestone in vaccinology comparable to smallpox eradication may be around the corner for paralytic polio, a crippling disease targeted for eradication in the near future. A campaign spearheaded by the World Health Organization has relied on massive immunization programs to carry out this objective. The project was shrouded by resistance in certain areas based on rumors that immunization causes sterility in male children. The regional resurgence in polio cases in certain areas of Asia and Africa resulting from this resistance is a setback, but one that can be overcome by education and observation of the benefits of vaccination. The fact that polio is not a threat worldwide and has been eliminated in most countries is a chunk that should not be overshadowed by delays in eradication program. In the United States and other industrialized nations, vaccines have eliminated a host of childhood diseases that were considered a part of growing up just 50 years ago. Measles, mumps, whooping cough or pertussis, tetanus, diphtheria, and polio are extremely rare or non-existent because of current vaccination practices, and can barely estimate the savings to society resulting from the prevention of these diseases. Aside from suffering and mortality, the cost to treat these illnesses and their after effects are sequelae such as paralysis, deafness, blindness, and mental retardation is immense, and dwarfs are course of immunization. For some diseases, immunization is the best, if not the only effective defense, because few antiviral drugs are now available. The major defense against influenza must be an effective vaccine. If an influenza pandemic recurs, as predicted by many experts, there will be a race between its spread and the manufacture and administration of an effective vaccine. At the time of this writing, much attention was being paid to an emerging strain of avian influenza. About 200 cases of infection in humans have been documented, of which about half were fatal. If this virus adapts to spread efficiently in humans, a major pandemic will result. Without a preventative vaccine, the devastation caused by the influenza pandemic of 1918, which left as many as 50 billion dead, will be equaled or surpassed. Despite the record of successful vaccines and our relies on them, opponents to vaccination programs have made claims that vaccines do more harm than good, and that childhood vaccination should be curtailed or even eliminated. There is no dispute that vaccines represent unique safety issues, since vaccines are given to people who are healthy. Furthermore, there is a general agreement that vaccines must be regulated and the public must have access to clear and complete information about them. Although the claims of critics must be evaluated, many can be answered by careful and objective examination of records. A recent example is the claim that the mercury containing preservative thimerosal, previously used in some vaccine preparations, causes autism and was responsible for recent increase in the incidence of the disorder, which is marked by intense self-absorption and inability to relate to others. So, autism generally manifests itself between 1 and 2 years of age, the time window in which many vaccines are given. The Danish government maintains meticulous health records on its citizens, a source of data that throws revealing light on the putative causal connection between thimerosal and autism. The records indicated that incidence of autism increased significantly since 1992 in Denmark. However, the records also showed that the use of thimerosal as a vaccine preservative had already been stopped completely in that country several years earlier. Data such as this make it difficult to link autism to the use of thimerosal and suggest the need for further research into the cause for the increase in autism. Perhaps the greatest current challenges in vaccine development is the lack of effective vaccines for major killers such as malaria and AIDS. It is hoped that the immunologists of today, using the tools of molecular and cellular biology, genomics, and proteomics, will make inroads into preventing these diseases. Further issue in vaccines is the fact that millions of children in developing countries die from diseases that are fully preventable by available safe vaccines. High manufacturing costs, instability of the products, and delivery problems keeping these vaccines from those they could greatly benefit. This problem could be alleviated in many cases by development of future generation vaccines that are inexpensive, heat stable, and administered without a needle. Then early studies of humoral and cellular immunity. Pasteur proved that vaccination worked, but he did not understand how. The experimental work of Emil von Behring and Shibasaburo Kitasato in 1890 gave the first insights into the mechanism of immunity. Erling von Behring and Nobel Prize in Medicine in 1901. Von Behring and Kitasato demonstrated that serum, the liquid non-cellular component recovered from coagulated blood from animals previously immunized to diphtheria, could transfer the immune state to unimmunized animals. In search of the protective agent, various researchers during the next decade demonstrated that an active component from immune serum could neutralize toxins, precipitate toxins, and agglutinate or clump bacteria. In such case, the active agent was named for the activity it exhibited: antitoxin, precipitin, and agglutinin respectively. So initially, different serum components were thought to be responsible for each activity, but during 1930s, mainly through the effects of Elvin Cabat, a fraction of serum, first called gamma globulin, now immunoglobulin, was shown to be responsible for all these activities. The active molecules in the immunoglobulin fraction are called antibodies. The term antibody and immunoglobulin may be used interchangeably, but usually the term antibody is reserved for immunoglobulins with non-specificity for an antigen. So because immunity was mediated by antibodies contained in body fluids known at the time as humors, it was called humoral immunity. The observation of von Behring and Kitasato was applied to clinical practice prior to the advent of antibiotic therapy for infectious diseases. Antisera, often prepared in horses, were given to patients with a variety of illnesses. Today, there are therapies that rely on transfer of immunoglobulins, and with the development of monoclonal antibody technology, antibody therapy is now a well-established commercial enterprise. So, emergency use of sera containing antibodies against snake or scorpion venoms for bite victims is a common practice. Whereas a vaccine is said to engender active immunity in the host, transfer of antibody with a given specificity confers passive immunity. Furthermore, there is a general agreement that vaccines must be regulated. So, we are talking about sera, active immunity in the host. Transfer of antibody with a given specificity confers passive immunity. Newborn infants benefit from passive immunization conferred by the presence of maternal antibodies in their circulation. Passive immunity may be given as a preventative or prophylaxis to those who anticipate exposure to given disease or to those with compromised immunity. Antisera to certain bacterial toxins such as diphtheria or tetanus can be administered to infected individuals to stop the disease caused by the toxin. A dramatic vignette of this application was the relay of dog sleds racing over 674 miles of frozen Alaskan landscape to bring diphtheria antitoxin from Fairbanks to the affected children in the ice-bound city of Nome during a diphtheria outbreak in 1925. The successful outcome is commemorated annually in the Iditarod dog sled race, and there is a state of Plato, one of the lead dogs used for the final leg of the delivery in New York's Central Park. In 1883, even before the discovery that a serum component could transfer immunity, Eli Mechnikov demonstrated that cells also contribute to the immune state of an animal. He observed that certain white blood cells, which he termed phagocytes, ingested or phagocytosed microorganisms and other foreign materials. Nothing that these phagocytic cells were more active in animals that had been immunized. Mechnikov hypothesized that cells, rather than serum components, were the major effectors of immunity. The active phagocytic cells identified by Mechnikovs were highly blood monocytes and neutrophils. And some major aspects related to theoretical challenges. Whereas development of safe and effective vaccines and the use of passive immune therapy remain challenging problems in clinical, also called translational research. The study of immunology also raised theoretical questions that occupied the scientific community. One question that surfaced early involved the relative roles of cellular and humoral immunity. The controversy developed between those who held to the concept of humoral immunity and those who agreed with Mechnikov's concept of cell-mediated immunity. It is now obvious that both are correct. The full immune response required both cellular and humoral responses. Early studies of immune cells were hindered by the lack of generally defined animal models and modern tissue culture techniques, whereas studies with serum took advantage of the ready availability of blood and established biochemical techniques to purify protein mediators of humoral immunity. Information about cellular immunity therefore lagged behind the unraveling of humoral immunity. In a key experiment in 1940s, Merrill Chase worked at the Rockefeller Institute succeeded in transferring immunity against the tuberculosis organism by transferring white blood cells between guinea pigs. Until that point, attempts to develop an effective vaccine or antibody therapy against tuberculosis had met with failure. This Chase demonstration helped to rekindle interest in cellular immunity with the emergence of improved cell culture techniques in the 1950s. The lymphocyte was identified as the cell responsible for both cellular and humoral immunity. Soon thereafter, experiments with chickens pioneered by Bruce Glick at Mississippi State University indicated that there are two types of lymphocytes: T lymphocytes derived from thymus mediated cellular immunity and B lymphocytes from the bursa fabricis, outgrowth of the cloaca birds, were involved in humoral immunity. The controversy about the roles of humoral and cellular immunity was resolved when the systems were shown to be intertwined. It became clear that both systems were necessary for the immune response. And one of the great enigmas confronting early immunologists was the specificity of the antibody molecule for foreign material and antigen, the general term for a substance that binds with a specific antibody. Jules Bordet at the Pasteur Institute expanded the concept of immunity beyond infectious diseases by demonstrating that non-pathogenic substances such as red blood cells from other species could serve as antigens. Serum from an animal inoculated with non-infectious material would nevertheless react with the injected material in a specific manner. The work of Karl Landsteiner and those who followed him showed that injecting an animal with almost any organic chemical could induce production of antibodies that could bind specifically to the chemical. These studies demonstrated that antibodies have a capacity for an almost unlimited range of reactivity, including response to compounds that had only recently been synthesized in the laboratory and had not previously existed in nature. In addition, it was shown that molecules differing in the smallest detail could be distinguished by their reactivity with different antibodies. Two major theories were proposed to account for this specificity: the selective theory and the instructional theory. The earliest conception of the selective theory dates to Paul Ehrlich in 19. In an attempt to explain the origin of serum antibody, Ehrlich proposed that cells in the blood expressed a variety of receptors, which he called side chain receptors, that could react with infectious agents and inactivate them. Or a concept used by Emil Fischer in 1894 to explain the interaction between an enzyme and its substrate. Ehrlich proposed that binding of the receptor to an infectious agent was like the fit between lock and key. Ehrlich suggested that interaction between an infectious agent and a cell-bound receptor would induce the cell to produce and release more receptors with the same specificity. According to Ehrlich's theory, the specificity of the receptor was determined in the host before its exposure to antigen, and the antigen selected the appropriate receptor. Ultimately, all aspects of Ehrlich's theory would be proven correct, with the minor exemption that the receptor exists as both a soluble antibody molecule and a cell-bound receptor, and soluble form is secreted rather than the bound form released. In the 1930s and 1940s, the selective theory was challenged by various instructional theories, in which antigen played a central role in determining the specificity of the antibody molecule. According to the instructional theories, a particular antigen would serve as a template around which antibody would fold. The antibody molecule would thereby assume a configuration complementary to that of the antigen template. This concept was first postulated by Frederick Brainel and Felix in about 1930 and redefined in the 1940s in terms of protein folding by Linnaeus pudding. The instructional theories were formally disproved in 1960s, by which time information was emerging about the structure of protein, RNA and DNA that would offer new insights into the vexing problem of how an individual could make antibodies against anything, almost anything. In the 1950s, selective theories resurfaced as a result of new experimental data and through the insights of Niels Jern, David Thalmade, and F. McFarland Burnett were refined into a theory that came to be known as clonal selection theory. Clonal selection. According to this theory, an individual lymphocyte expresses membrane receptors that are specific for a distinct antigen. This unique receptor specificity is determined before the lymphocyte is exposed to the antigen. Binding of antigen to its specific receptor activates the cell, causing it to proliferate into a clone of cells that have the same immunological specificity as the parent cell. The clonal selection theory has been further refined and is now accepted as an underlying paradigm of modern immunology. Then infection and immunology, along with the developing field of immunology, grew the study of medical microbiology, which covered the identification of infectious agents and their modes of causing disease. Organisms cause disease are termed pathogens, and their means of attacking the host is called pathogenesis. The human pathogens can be grouped into major categories: viruses, bacteria, fungi, and parasites. Of major interest, there are the means by which effective immunity to the pathogens can be achieved. An effective defense relies heavily on the nature of the individual microorganisms. For example, because viruses require mammalian cells for their replication and multiplication, an effective defense strategy may involve recognizing and killing any cell that is virus infected before the life cycle of virus can be completed. For organisms that replicate outside host cells, prompt recognition of these invaders by antibodies or soluble molecules can be used, followed by cellular and molecular immune mechanisms that eliminate the pathogen. Some pathogens that pervade our environment cause no problem for healthy individuals because there is adequate pre-existing immunity. However, individuals with deficiencies in immune function may be susceptible to diseases caused by these ubiquitous microbes. For example, the fungus Candida albicans, present nearly everywhere, cause no problem for most individuals. However, in the presence of lowered immunity, it can cause an irritating rash and a spreading infection in the mucosa lining of the oral and vaginal cavities. The rash, called thrush, may be the first sign of immune dysfunction. If left unchecked, this can spread, causing systemic candidiasis, a life-threatening condition. Candidiasis. Another example is the virus herpes virus simplex, which normally causes small lesions around the lips of the genitalia. In those with deficit immunology, these lesions may spread to cover large portions of the body. Such infections by ubiquitous microorganisms, often observed in case of immune deficiency, are termed opportunistic infections. Several rarely seen opportunistic infections identified in early AIDS patients were the signal that the immune system of these patients were seriously compromised. For some pathogens known to cause serious diseases, the means to achieve immunity as well documented and allow control of the disease. For example, tetanus, commonly called lockjaw, is caused by common soil bacterium Clostridium tetani, which acts through a toxin that attacks the nerve system or neurotoxin. So, untreated tetanus leads to death within a short time. For some pathogens not to cause serious disease, the means to achieve immunity as well documented and allow control of the disease. For example, tetanus, commonly called lockjaw, is caused by a common soil bacterium, Clostridium tetani, which acts through a toxin that attacks the nerve system, neurotoxin. Untreated, tetanus leads to death within a short time. There is an effective vaccine to tetanus, and should that fail, antibodies to the toxin can be administered to offset the potentially fatal disease. Prior to the availability of these prevention and treatment measures, anyone suffering a puncture wound by a dirty object such as a rusty nail was at a risk of fatal tetanus infection. The marked contrast to the success in controlling tetanus is the failure to devise immune strategies to control HIV infection, which leads to AIDS. The immune system must deal with all types of pathogens and has evolved multiple strategies for dealing with invasion by a pathogen that has passed the first barrier of skin and mucosa. We will see that some of these strategies are poised to respond the instant a pathogen breaches the host's barriers. Other defenses are crafted to act other infections is established. Innate and adaptive immunity. Immunity, the state of protection from infectious disease, has both a less specific and a more specific component. The less specific component, innate immunity, provides the first line of defense against infection. Most components of innate immunity are present before the onset of infection and constitute a set of disease resistance mechanisms that are not specific to a particular pathogen but include cellular and molecular components that recognize classes of molecules peculiar to frequently encountered pathogens. The first hurdle for a pathogen involves breaking the barriers that protect the host. Obvious barriers include the skin and the mucosal membrane, the acidity of the stomach contents, and of perspiration poses a further barrier to organisms unable to grow in acidic conditions. Enzymes such as lysozyme, which is present in tears, attack the cell walls of certain bacteria in contact. The importance of these barriers become obvious when they are breached. Bites of animals or insects puncture the skin and can induce a number of diseases. Animal bites can communicate rabies or tetanus, whereas insects carry a host of diseases including malaria from mosquitoes, plague from fleas, and Lyme disease from ticks. A dramatic example of barrier loss is seen in burn victims who lost the protective skin at the burn site and must be treated aggressively with drugs to prevent bacterial and fungal infection. Beyond the primary barriers, innate immunity includes the host of cells such as phagocytes demonstrated by Mechnico as well as antimicrobial compounds synthesized by the host that can recognize and neutralize invaders based on common molecular surface markers. And phagocytic cells are a barrier to infection. An important innate defense mechanism is the ingestion of extracellular particulate material by phagocytosis. So phagocytosis is one type of endocytosis, the general term for the uptake by a cell of material from its environment. In phagocytosis, a cell's plasma membrane expands around the particulate material, which may include whole pathogenic microorganisms. Most phagocytosis is conducted by specialized cells such as blood monocytes, neutrophils, and tissue macrophages. Most cell types are capable of another forms of endocytosis, such as receptor-mediated endocytosis, in which extracellular molecules are internalized after binding to specific cellular receptors, such as pinocytosis, the process by which cells take up fluid from the surrounding medium along with any molecules contained in it. And soluble molecules contribute to innate immunity. A variety of soluble factors contribute to innate immunity, among them the protein lysozyme, the interferon protein, and components of the complement system. Lysozyme, a hydrolytic enzyme found in mucus secretions and in tears, is able to cleave the peptidoglycan layer of the bacterial cell wall. So, interferon comprises a group of proteins produced by viral infected cells. Among the many functions of interferons is the availability to bind to nearby cells and induce a generalized antiviral state. Complement examined includes a group of serum proteins that circulate in an inactive state. A variety of specific and non-specific immunologic mechanisms can convert the inactive forms of complement proteins into an active state with the ability to damage the membranes of pathogenic organisms, either destroying the pathogens or facilitating their clearance. So, complement occupies a position that truly straddles the innate and adaptive immune systems, in that certain components may directly deal with pathogens, whereas others require prior binding of antibodies to activate its effector system. So, reactions between complement molecules or fragments of complement molecules and cellular receptors trigger activation of cells of the innate or adaptive immune system. Recent studies on collections indicate that these proteins may kill certain bacteria directly by disrupting their lipid membranes, or alternatively by aggregating the bacteria to enhance their susceptibility to phagocytosis. Many of the molecules involved reactions between complement occupies a position that truly straddles the innate and adaptive immune systems, in that certain components may directly deal with pathogens, whereas others require prior binding of antibodies to activate its effector system. Reactions between complement molecules or fractions of complement molecules and cellular receptors trigger activation of cells of the innate or adaptive immune systems. Recent studies on collections indicate that these proteins may kill certain bacteria directly by disrupting their lipid membranes, or alternatively by aggregating the bacteria to enhance their susceptibility to phagocytosis. Many of the molecules involved in innate immunity have the property of pattern recognition, the ability to recognize a given class of molecules. Because certain types of molecules are unique to microbes and never found in multicellular organisms, the ability to immediately recognize and combat invaders display such molecules is a strong feature of innate immunity. Molecules with pattern recognition ability may be soluble, like lysozyme and the complement components described above, or they may be cell-associated receptors, such as those designed in the toll-like receptors described later. So, should be invading pathogens breach the host's physical and chemical barriers. It may then be detected by pattern recognition molecules of the host and taken up by phagocytic cells, causing the system to react with an inflammatory response. This response concentrates elements of innate immunity at the inflammation site and may result in the maskling of specific immune response against the invader. The specific response called upon by the inflammation in the adaptive, sometimes called acquired immune response. In contrast to the broad reactivity of the innate immune systems, which is uniform in all members of a species, the specific component adaptive immunity does not come into play until there is a recognized antigenic challenge to the organism. Adaptive immunity responds to the challenge with a high degree of specificity as well as the remarkable property of memory. Typically, there is an adaptive immune response against an antigen within 5 or 6 days after the initial exposure to that antigen. Future exposure to the same antigen results in a memory response. The immune response to the second challenge occurs more quickly than the first, is stronger, and is often more effective in neutralizing and clearing the pathogen. The major agents of adaptive immunity are lymphocytes and antibodies that produce innate adaptive immunity. Because adaptive immunity response requires some time to marshal, innate immunity provides the first line of defense during the critical period just after the host exposure to the pathogen. In general, most of the microorganisms encountered by a healthy individual are readily cleared within a few days by defense mechanisms of the innate immune system before they activate the adaptive immune system. So, collaboration between innate and adaptive immunity increase immune responsiveness. It is important to appreciate that the innate and adaptive immune systems do not operate independently. They function as a highly interactive and cooperative system, producing a combined response more effective and either branch could produce by itself. Certain cellular and molecular immune components play important roles in both types of immunity. An example of cooperation is seen in the encounter between macrophages and microbes. Interaction between receptors on macrophages and microbial components generate soluble proteins that stimulate and direct adaptive immune responses, facilitating the participation of the adaptive immune system in the elimination of the pathogen. The soluble proteins are growth factor like molecules known by the general name cytokines. The cytokines react with receptors on various cell types and signal the cell to perform functions such as synthesis of new factors or to undergo differentiation to a new cell type. A restricted cell of cytokines have chemotactic activity and recruit specific cells to the site of cell secreting that cytokine. These are called chemokines. The type of intracellular communication mediated by cytokines is referred to by the general term signaling. So basically, signaling involves the reaction between a soluble molecule ligand and a cell membrane bound molecule receptor, or between membrane bound molecule to on two different cells. The interaction between receptor and its ligand leads to metabolic adaptations in cells. There are an enormous number of different signal transduction pathways, all exhibiting common themes typical of these interactive processes. Signal transduction begins when a signal binds to its receptor. Receptors may be inside or outside the cell. Signals that cannot penetrate the cell membrane bind to receptors on the cell surface. This group includes water-soluble signaling molecules and membrane-bound ligands, MHC peptide complexes, for example. Then hydrophobic signals such as steroids can diffuse through the cell membrane and are bound by intracellular receptors. Many signal transduction pathways involve the signal-induced assembly of pathway components. Molecules known as adapter proteins bind specifically and simultaneously to two or more different molecules with roles in the signaling pathway, bringing them together and promoting their combined activity. So we will learn about an example of cooperation is seen in the encounter between macrophages and microbes. We learned about cytokines. Then signal transduction begins when a signal binds to its receptor. Receptors may be inside or outside the cell. Signals that cannot penetrate the cell membrane bind to receptors on the cell surface. This group includes water-soluble signals. This group includes water-soluble signaling molecules and membrane-bound ligands like MHC peptide complexes, for example. Then hydrophobic signals such as steroids can diffuse through the cell membrane and are bound by intracellular receptors. Many signal transduction pathways involve the signal-induced assembly of pathway components. Molecules known as adaptive proteins bind specifically and simultaneously to two or more different molecules with roles in the signaling pathway, bringing them together and promoting their combined activity. Signal receptor often leads to the generation within the cell of a second messenger, a molecule or ion that can diffuse to other sites in the cell and stimulate metabolic changes. Examples are cyclic nucleotides CAMP, CGMP, calcium ion Ca2+, and membrane phospholipid derivatives such as diacylglycerol DAG and inositol triphosphate IP3. Protein kinase and protein phosphatases are activated or inhibited. Kinase catalyze the phosphorylation of target residues tyrosine, serine, or threonine of key signal transduction proteins. So phosphatases catalyze dephosphorylation, reversing the effect of kinases. The enzymes play essential roles in many signal transduction pathways of immunological interest. Signals are amplified by enzyme cascades. An enzyme is a signaling pathway. Once activated, catalyzes many additional reactions. The enzymes may generate many molecules of the next component in the pathway. Activate many copies of the next enzyme in the sequence. This greatly amplified amplify the signal at each step, and offer opportunity to modulate the intensity of the signal. So these processes are very important. An activity that results from single transaction may be synthesis or secretion of certain proteins, differentiation or the initiation or cessation of specific functions. For example, stimulated macrophages secrete cytokines that can direct adaptive immune response of lymphocytes against specific pathogens. In a complementary fashion, the adaptive immune system produces signals and components that increase the effectiveness of immune response. Some T cells, when they encounter appropriately presented antigens, synthesize and secrete cytokines and increase the ability of macrophages to kill the microbes they have ingested. Also, antibodies produced against an invader bind to the pathogen, marking it as a target for attack by macrophages or by complement proteins and serving as a potent activator of the attack. A major difference between adaptive and innate immunity is the rapidity of the innate immune response, which utilizes a pre-existing but limited repertoire of responding components. Adaptive immunity compensates for its slower onset by its ability to recognize a much wider repertoire of foreign substances, and also by its ability to improve during a response, whereas innate immunity remains constant. Adaptive immunity is highly specific. Adaptive immunity is capable of recognizing and selectively eliminating specific foreign microorganisms and molecules, foreign antigens, unlike innate immune responses. So signaling pathways are initiated when a signal binds its receptor. Ligand binding to receptor induces assembly of signaling pathway components. Generation of second messenger carries signal to cell interior. Phosphorylation, dephosphorylating cycles under control of the signaling pathway deactivate additional pathway components. Then enzyme cascades amplify the signal, converting the molecules to their active forms. And adaptive responses are not the same in all members of species, but are reactions to specific antigenic challenges. Adaptive immunity display 4 characteristic attributes: antigenic specificity, diversity, immunological memory, self non-self recognition. The antigenic specificity of the adaptive immune system permits it to distinguish subtle differences among antigens. Antibodies can distinguish between 2 protein molecules that differ in only a single amino acid. The immune system is capable of generating tremendous diversity in its recognition molecules, allowing it to recognize billions of unique structures on foreign antigens. This ability is in contrast to the pattern recognition molecules of the innate system, which recognize broad classes of organisms based on molecular structures present on them. The adaptive system can recognize a single type of organism and differentiate among those with minor genetic variations. Once the adaptive immune system has recognized and responded to an antigen, it exhibits immunologic memory. That is, the second encounter with the same antigen induces a highlighted state of immune reactivity. Because of this attribute, the immune system can confer lifelong immunity to many infectious agents after an initial encounter. Finally, the immune system normally responds only to foreign antigens, indicating that it is capable of non-self recognition. The ability of immune system to distinguish self from non-self and respond only to non-self molecules is essential. As described below, failure of the ability to distinguish self from non-self leads to an inappropriate response to self components and can be fatal. So lymphocytes and antigen-presenting cells cooperate in adaptive immunity. An effective immune response involves two major groups of cells: lymphocytes and antigen-presenting cells. Lymphocytes are one of the many types of white blood cells produced in the bone marrow by the process of hematopoiesis. Lymphocytes leave the bone marrow, circulate in the blood and the lymphatic systems, and reside in various lymphoid organs. Because they produce and display antigen-binding cell surface receptors, lymphocytes mediate the defining immunologic attributes of specificity, diversity, memory, and self-non-self recognition. The 2 major populations of lymphocytes, B lymphocytes and T lymphocytes are described briefly, and later we will be describing these topics elaborately. Okay. So B cell, the B lymphocytes mature in the bone marrow. On release, each express a unique antigen binding receptor on its membrane. This antigen binding or B cell receptor is a membrane bound antibody molecule. Membrane bound antibody molecule. Antibodies are glycoproteins that consist of two identical polypeptides called the heavy chains and two shorter identical polypeptides called the light chains. Each heavy chain is joined to a light chain by disulfide bonds, and the heavy or light chains pairs are linked together by additional disulfide bonds. The amino terminal ends of the pairs of heavy and light chains form a site to which antigen binds. When a naive B cell, one that has not previously encountered antigen, first encounters the antigen that matches its membrane bound antibody, the binding of the antigen to the antibody causes the cell to divide rapidly. Its progeny differentiate into memory B cells and effector B cells called plasma cells. So memory B cells have a longer lifespan than naive cells, and they express the same membrane bound antibody as their parent B cell. So plasma cells produce the antibody in the form that can be secreted and have little or no membrane bound antibody. Although plasma cells live for only a few days, they secrete enormous amounts of antibody during this time. A single plasma cell can secrete hundreds to thousands of molecules of antibody per second. Secreted antibodies are the major effector molecules of humoral immunity. Then next, T lymphocytes. A quick recap about T lymphocytes. T lymphocytes also arise in the bone marrow, unlike B cells which mature in the marrow. T cells migrate to the thymus gland to mature. Maturing T cells express a unique antigen binding molecule, the T cell receptor, on their membrane. There are two well defined subpopulations of T cells, T helper cells and T cytotoxic T cells. So T helper and T cytotoxic cells can be distinguished from one another by the presence of their CD4 or CD8 membrane glycoproteins on their surface. T cells displaying CD4 are generally known as TH cells, whereas those TH in the sense helper. Okay. Whereas those displaying CD8 generally function as a TC cell. TC in the sense cytotoxic T cells. I recently categorized third types of T cells called a T regulatory cell carries CD4 on its surface but may be distinguished from T4 and TC cells by cell surface markers associated with its stage of activation. Unlike membrane-bound antibodies on B cells, which recognize a free antigen, most T cell receptors can recognize only antigen that is bound to cell surface proteins called the major histocompatibility complex (MHC) molecules. MHC molecules are polymorphic, genetically diverse glycoproteins found in cell membranes. These are two major types of MHC molecules: MHC class 1 molecules, which are expressed by nearly all nucleated cells of vertebrate species, and class 2 MHC molecules, which are expressed only by antigen presenting cells or APCs. When a new T cell encounters antigen combined with MHC molecule on a cell, the T cell proliferate and differentiate into memory T cell and various effector T cells. After a TH cell recognizes and interacts with an antigen MHC class 2 molecule complex, the cell is activated. It undergoes a metabolic transformation and begins to secrete various cytokines. The secreted cytokines play an important role in activating B cells, T cells, macrophages, and various other cells that precipitate in the immune response. Difference in the type of cytokines produced by activated TH cells result in various patterns of immune response. And possible response is the introduction of a change in TC cells to form cytotoxic T lymphocytes that exhibit cell killing or cytotoxic activity. CTLs have a vital function in monitoring the cell of the body and eliminating any that display antigen, such as virus-infected cells, tumor cells, and cells of a foreign tissue graft. Other topics related to this, like antigen-presenting cells interact with T cells, humoral and cellular immune response exhibit different effector functions, and antigen receptors of B and T lymphocytes. Some other aspects related to the major histocompatibility complex molecules bind antigenic peptides. Antigen selection by lymphocytes cause clonal expansion immune dysfunction and its consequences. These topics